Video is about nucleic acids, so DNA and RNA being the main two. Um, so structurally, right, how is it that these molecules are information, right? That's really what they are. They contain the sugar ribose, which was covered in the carbohydrate video. They also contain a nitrogenous base. So when we make the subunit, it has these three pieces. So we have a ribose sugar, a nitrogenous base, and a phosphate group. So the difference between RNA and DNA is whether the ribose is deoxyribose or just simply ribose, and the nitrogenous bases. So in DNA we find adenine, thymine, cytosine, and guanine, so A's, T's, C's, and G's. And in RNA, instead of thymine, we find uracil, U. All right, that's RNA. All right, so just some small minor differences there. Okay, so this is DNA, or a nucleotide to make DNA. We know this because here at the 2 we don't have a hydroxyl group, and therefore it is deoxyribose. Okay, and so DNA is deoxynucleic acid. All right, our nucleobase, or nitrogenous base, here is adenine, and we have a phosphate group here. Okay, so those are kind of the basic components of our monomer that we're going to make the DNA polymer from. So we have two different types of nitrogenous bases. We have the purines, which have a double ring, right? So adenine and guanine, and we have the pyrimidines, which are a single ring, and they are here. You do not need to memorize these structures. Simply know them as A's, T's, C's, and G's, and uracil, so U's. Okay. Now, let's look at how we would make this polymer or put this polymer together. All right. So let's look at this top nucleotide. All right. So our base here is guanine, so there's our nucleobase. It's attached at the 1 carbon of our ribose sugar, which is a 5-carbon sugar. Then at the 2, 3, 4, 5, at the 5 position, we have our phosphate group hanging off the end here. Okay, so we refer to this as our 5' phosphate. Now, to link these nucleotides together, we link the 5 carbon of one ribose to the 3 carbon of another. All right, and that is done via the phosphorus group. Okay, so we call this red highlighted square the sugar phosphate backbone, and the linkage between these two is a phosphodiester bond. Okay, phosphodiester bond. Now, at this other end of our molecules, if you look down here at the bottom, we count our carbons on our ribose, 1, 2, 3. We have a hydroxyl group here. That's our 3' hydroxyl group. So we often refer to DNA as being directional, 5' to 3', 3' to 5', and so on. All right? So just keep that in mind in terms of the orientation of this molecule. Okay, so what we want to do is, let's start over here on the right. And this is the alpha helix, right? So this is the double helix. So we've got 2 strands of DNA running parallel to each other, or what we call anti-parallel, because one of them is going from a 5' end to a 3' end, and the other is going 3' at the top to 5' at the bottom. All right, so they run anti-parallel, kind of like a highway, right? The highway goes anti-parallel depending on whether you're heading north or south, for example, on that highway. Okay, so let's zoom in and get a better look at this. And what I want you to see is, we've got a 5' end on the left, right? So our red sort of strand, our blue strand is 3' at the top, 5' at the bottom, and you'll see how they've actually made everything upside down on the right, because they are actually running in opposite directions to each other. Okay? So some things that need to occur, we have to have complementary base pairing. So on the inside, the nitrogenous bases hydrogen bond to each other. So it's always an A to a T and a C to a G, or a G to a C, however you want to think of it. But that's how you make the proper bonding across the between the 2 sugar backbones. All right. The C to G bond has 3 hydrogen bonds. The A to T has 2 hydrogen bonds. So if we zoom in here, we can see our guanine cytosine. Here's our 3 hydrogen bonds. Here's our thymine adenine, 2 hydrogen bonds. And here we have our phosphodiester linkage hooking up our sugar backbone. All right. Now, why A to T and G to C? Think of it, if you were building a spiral staircase, you would need it to fit specific dimensions. Like, let's say that you had a 3-foot wide staircase. Well, A to T gives you 3 feet. C to G gives you 3 feet. If you do G to A, you get 5 feet. If you do T to C, you get 2 feet, right? So it doesn't work. So the dimensions of this thing have to be perfect, and therefore the bonding and the base pairing is exact. And so we call this complementary base pairing, G to C, A to T. The strands run anti-parallel to each other in the 5' to 3' direction. Now, let's look at ATP, which is an energy useful molecule in the cell. It's really what we often refer to as the energy currency of a cell. And the energy is in these high energy bonds here in these phosphate groups. All right, so adenosine triphosphate, adenosine diphosphate, adenosine monophosphate refers to how many of these phosphate groups are here. And all of when we build DNA or we build RNA, we use the triphosphate form and we break those 2 phosphate groups off to make our phosphodiester bond. All right? So, in comparison to proteins, the DNA has 4 different nitrogenous bases. So we can basically make a pattern of A's, T's, C's, and G's to store the information. All right? Proteins are extremely complex. They're not good for storing protein because they are too complex. All right? And we'll get into that a bit more later.